Good morning, students. Today let me explain you about a new concept, what is called as sex determination system, or in other words sex determination mechanism. And this concept is the last part of that chapter, sex linked inheritance chapter. Now the next one is, what is sex determination? Sex determination is the process of sex differentiation by which whether a particular individual will develop into male or female sex. And the expression of the sex is governed by chromosomes and genes. And in unisexual animals, chromosomes are of two types, as you know. They are autosomes and allosomes. What are autosomes? The chromosomes which do not differ in morphology and number in male and female. That means they are unique to both male and female of a particular species. And generally these autosomes are also called as somatic chromosomes or vegetative chromosomes, as they carry the genes which are responsible for most of the somatic characters or the vegetative characters of that particular organism. Then allosomes are the sex chromosomes, and the chromosomes which differ in morphology and number in male and female and contain genes that determine the sex. Such chromosomes are called allosomes or sex chromosomes. This we have already discussed in brief under sex-linked inheritance. And these unisexual diploid individuals have two sex chromosomes, and rest of them are autosomes. And let us go through some differences between autosomes and allosomes. Autosomes refers to the chromosomes other than sex chromosomes, and they do not determine the sex of individuals. Whereas allosomes or sex chromosomes, they are the sex chromosomes that determine the sex of individuals. And chromosome number is 2n-1 in case of autosomes, and in case of allosomes the chromosome number is 2 only. That means in autosomes there will be 2n number, that is 2 sets of chromosomes are present, whereas in case of allosomes only 2 chromosomes are present, either XX or XY or some other pattern of sex chromosomes. And the morphology is similar in male and female with reference to autosomes. There is no change in terms of morphology or in terms of number also. Whereas in case of allosomes, the morphology is different in male and female. And the number differs from species to species, that is with reference to autosomes, whereas each diploid organism has usually 2 allosomes only. And autosomes do not exhibit sex linkage, whereas allosomes exhibit sex linkage, which we have already discussed in the previous concept, that is sex-linked inheritance, where we have discussed about the patterns of sex-linked inheritance with reference to the hemophilia and color blindness in case of man and eye color in case of Drosophila. Coming to sex characters. The primary characters are being determined by the gametes, whereas the secondary characters are by genitalia, especially the hormones, that is the sex hormones produced by the respective individuals. Coming to chromosomal sex determination. In a diploid individual, there are 2n number of chromosomes, that is 2 autosomes and 2 sex chromosomes. It is actually 2 sets of autosomes and sex chromosomes. There are 3 types of chromosomal sex determination. They are sex determination by allosomes, second one Diploid-haploid system of sex determination, and third one Genic balance system. Now let us go through them one by one in detail. Coming to the first one, allosomal sex determination. Allosomes are sex chromosomes that are generally of X and Y types, but in some birds they are of Z and W types, and so also in some insects they have ZZ ZW mechanism. That we'll discuss it later. And the sex with similar type of sex chromosomes, that is XX, is known as homogametic sex, and with dissimilar type of sex chromosomes, that is XY, as heterogametic sex. There are four different systems of allosomal sex determination. They are XXXY, that is female male system. Second one, XXX0, that is female male system. And X0XX, that is again female male system. And ZWZZ, which is female male system. So these are the four types of allosomal sex determination. Coming to the types of sex chromosomal mechanism of sex determination, the one is heterogametic males, the second type is heterogametic females, and heterogametic males includes XXX0 type and XXXY type, which we have seen in the previous slide. So also we have seen in the previous slide that there are some females which are said to be heterogametic, and the types are Z0 ZZ system, ZW ZZ system. See, this is what with reference to the two types of sex chromosomal mechanism of determination. Now the first one, XXXY, that is female male system, and this type is seen in Drosophila, man, and some other mammals. In this system, female has two X chromosomes, which is including in case of man, where it is said to be homogametic, as it produces only one type of gamete, that is every gamete is having the sex chromosome of X type, whereas the male has one X and one Y chromosome, because male is heterogametic, and it produces two types of gametes, that is one type of gamete with the X chromosome and one set of gametes with Y chromosomes. And in the union of X ovum with X sperm leads to the development of female sex, and if X ovum unites with Y sperm, it produces male sex. So it is very clear that wherever the chromosome configuration, particularly with reference to allosomal configuration, if it is XX type, then it will be female, and if it is XY, it is invariably producing male sex. So this is what about XXXY system, and it is referred to as female male system because XX denotes to female sex and XY denotes to male sex. Second type, XXX0, which is again a female male system, and this system is seen in grasshoppers and many Orthoptera and hemipteran insects. And in this system, female has double X chromosomes, that is XX, and male has single X chromosome, hence it is represented as X0. It is not XO, it is X0. And the female is again homogametic here, and it produces all the X with X chromosome only, whereas the male is heterogametic, which produces sperms, half of which have X chromosome and other half have none. And the union of egg with sperm having X chromosome will give rise to female sex, and with sperm having none results in development of male sex. So this pattern is said to be XX-X0 system. Then the third type, X0-XX system, which is said to be female male system or female heterogametic, and it is said to be female heterogametic because the female is having only one X chromosome, and it is found in very few species of insects like Fumea. Sorry. In this system, female has only one X chromosome, as I said, and hence it is heterogametic, whereas male is said to be homogametic here because male has two X chromosomes. And as a result of meiosis, 50% of the eggs of such female carry an X chromosome and remaining 50% carry none. That means they carry only autosomes but no sex chromosome. On the other hand, the male is homogametic and the male has two X chromosomes and produces all the sperms with one X chromosome only. That is, one X chromosome along with it some autosomes too. Thus, male sex is homogametic here. And the union of X sperm with ovum having X chromosome gives rise to male sex, and the union of X sperm with ovum having none leads to the development of female sex. So this is with reference to X0XX system, that is female heterogametic system. And the next type, ZWZZ type, which is again called female male system, and this is seen in birds, butterflies and moths, including silk moth. And here female is heterogametic and it produces two types of gametes, that is some with Z chromosome and some with W chromosome. Whereas male is homogametic and it produces all the sperms of the same type carrying one Z chromosome only, because male carries only two Z chromosomes. And the union of Z sperm with ovum having Z chromosome gives rise to male, and the union of Z sperm with ovum carrying W chromosome leads to the development of female sex. Then the next main type will be Diploid-haploid, that is female male system, and this is found in honeybees, ants and termites. In honeybees, the females have diploid, that is 2n number is equal to 32 chromosomes, and the drones or males have haploid, that is they have only one set of chromosomes, that is n is equal to 16 chromosomes. And the females are queen and workers, that is what we have already discussed in apiculture chapter in the previous semester. And the females which feed on the royal jelly develop into queen, while the queen is fertile and workers are sterile individuals, that is sterile females. And the queen produces haploid eggs. However, the haploid male bees produce haploid sperms by mitosis rather than by meiosis. And the union of egg with sperm gives rise to diploid larvae which become female. And here is the schematic representation of this system which is explained in a simple way. So at the beginning, as you see in the slide, the queen, if we are laying eggs, if there is fertilization of such eggs, it will produce into diploid larvae. Whereas if it is not fertilized, if the eggs are not fertilized, then they will produce haploid larvae, and that will develop into drones. So these drones are developed without the actual act of fertilization, the mechanism what we call it otherwise as parthenogenesis. So the drones, which are said to be the fertile males of the colony, they are being produced by parthenogenesis, that is without the fertilization act or process. And on the other hand, as we have studied here, if the eggs produced by the queen, if they are fertilized, they will produce 2n condition, and that will produce diploid larvae. And if the diploid larvae feed on royal jelly, they will develop into the queen, and if they are not feeding on the royal jelly, they will develop into worker bees, which are said to be sterile females. Whereas queen is the only fertile female of the colony. That is what we have already discussed in apiculture chapter. And the drone, as it is the fertile male of the colony, it will produce the sperms, and those sperms, if they are fused with the eggs, they will produce the 2n condition, that is zygote, and they will develop into diploid larvae. And the same mechanism, if the diploid larvae feed on royal jelly, they will develop into queen. If they are not feeding on the royal jelly, they will develop into worker bees. Coming to the next system, what is called as genic balance system. As the name itself suggests, it is the balance between the autosomes and the allosomes. Hence the system is called genic balance system. And this system was developed by Bridges in 1922 in Drosophila. And according to this theory, the sex of an individual is determined by a balance between the genes for maleness and those for femaleness present in the individual. And the genic balance theory states that the sex determining genes are present on both X chromosomes as well as on autosomes. So that is why there should be a balance between these autosomes and allosomes or the sex chromosomes, so that the sex is going to be manifested. And the male sex determining genes are present on autosomes generally, and female sex determining genes are present on the X chromosome. So this also you should remember here, that the male determining genes are present on autosomes, whereas the female determining genes are present on the allosomes. So the balance between the autosomes and allosomes here is going to determine the sex of the individual. The sex expression is determined by the balance of genes on autosomes and X chromosome. In other words, the expression of sex depends on the ratio of X chromosomes to that of autosomes. And this ratio is represented as X by A ratio. That is, X refers to the sex chromosome and A refers to autosomes. The genic balance is governed by X by A index. That is what we have seen in the previous slide itself. And the individuals with index of 1 develop into female, and those with sex index of 0.5 into male. It is quite natural here that you should observe one thing, that if the balance is towards 1, then it will develop into female. Whereas if the balance is the ratio is towards 0.5, then it will produce into male. And what happens if the ratio is ranging beyond or below this particular ratio? That we will discuss it in the next slide. If the sex index is between 1 and 0.5, the resulting individual will be neither a female nor a female, but have an intermediate sex expression and is called intersex. So it is very clear here that if it is exactly 1 ratio, or the X by A index giving you the value of 1 exactly, it will produce female, and 0.5 giving rise to male. And if it is between 1 and 0.5, the sex will be intersex, neither a male nor a female. The sex index of 1.5, which is higher than the sex index of the normal female, gives rise to super female. That is, if the index value goes above 1.5, it will produce super female. On the other hand, a sex index of 0.33, that is lesser than 0.5, which is lower than the sex index of the normal male, it gives rise to super male. So I hope you understood the mechanism here. The sex index value of 0.5 gives rise to male and 1 gives rise to female, and if it is between 0.5 and 1, it will be the it will be the intersex, and if it is above 1.5, super female, and below 0.5, it is super male. So that detailed mechanism of sex determination based on the X by A ratio or the value. It is being summarized here with the help of a tabular column. Let me explain a few of them. So coming to the ploidy, and this tabular column has the rows like this: ploidy, number of X chromosomes, number of autosomes, and number of the sex index ratio or value, and the expression of the sex. And if the individual is diploid, that is having 2n condition, and the chromosomes will be 3, that is X chromosome, and the number of autosomes is 2 only, so 3 by 2 will give you a ratio or the value of 1.5, so such individuals become super female. And if it is 4 number of sex chromosomes, that is in triploid condition, then the number of autosomes is 3, and index, that is sex index, will give you a value of 1.33. The individual become super female. And likewise, you can see the expression of the sex in Drosophila melanogaster with reference to this Genic balance theory that is being shown here in the tabular column, and that is for your reference. Now the sex index is calculated by the number of X chromosomes on the numerator and the number of autosomal sets in the denominator. So again, it is the repetition here, that is individuals with the sex index of 1.0 are normal female, that is XX diploid female, triple X triploid fly, and 4X condition, that is tetraploid condition. So all these will be female. And the flies having the sex index of 0.5 are normal males, that is X0 diploid flies, that is males, and XX00 tetraploid fly. Now coming to environmental sex determination, though it is not given over there in the syllabus, let me explain it in a brief way, because that is also a mechanism of sex determination in some organisms. So in some organisms, the sex determination, especially in case of the sea worm, Bonellia and Dinophilus, and even horsetail plant, what is called as equisetum, it is governed by environmental conditions and also includes some hormonal effects. In Bonellia, a sea worm, the larvae which remain free in the sea water and settle on the sea bottom are differentiated into females, while those larvae which settle on the proboscis of the female develop into tiny males. So very interesting mechanism here. What is that? This mechanism, that is environmental sex determination mechanism, explains in Bonellia larvae, those which are settled in the bottom are differentiated into females, okay, and those which are remain free in the sea water also, they will be turning out into females, while those larvae which settle on the proboscis region of the females develop into tiny males. So it's very clear here that those larvae which settles on the proboscis of the female, they will turn out into males. So environmental factors influences the expression of the sex here. Hence the mechanism is called the environmental sex determination. Now coming to sex mosaics in Drosophila. And sex mosaic. What is sex mosaic? It is a combination of male and female features in the body of the same individual, which is also called gynandromorph. So Drosophila flies, that is 1 in 2000, have male tissues in one part of the body and female tissues in other. It's a very unique condition found in Drosophila. So half of the body shows male characters and half of the body shows female characters. Hence the mechanism is called sex mosaic or gynandromorph. You know why it's called gynandromorph? The term itself says that gyn refers to or gyno refers to female sex and andro refers to male sex, especially the morphological types. So that is why the individuals are said to be the sex mosaics or gynandromorph. And individuals with such sex mosaics are also known as gynandromorphs or gynanders. So that is what is being explained here in this slide with the help of a photograph of a Drosophila. And as you see here, one part of that body of that fly is showing female characters and the other half showing the male characters. It's a very unique thing found in Drosophila. And there are three patterns of sex mosaics are found in Drosophila. They are as follows. Number 1, bilateral sex mosaic. As the name itself suggests, one side of the fly is male and the other side is female, and this is the most common type of sex mosaic seen in Drosophila. And the second type is antero-posterior sex mosaic, wherein the front half of the fly is one sex and the rear half of the other sex. So this type of gynandromorph is said to be antero-posterior sex mosaic or gynandromorph. The name itself suggests anterior part will be female and the posterior part will be male, or it is vice versa. Then sex mosaic in patches. Sometimes only a quarter of the body is male and rest is female, or just a small patch of tissues may be male and rest of the body is female. As the male characters and female characters are expressed in terms of patches in different regions of the body of such Drosophila, the mechanism is called sex mosaic in patches. However, these later two types are not generally seen. It is of very rare occurrence, and the most common occurrence will be bilateral sex mosaic, as we have seen in the previous slide with the help of a photograph of a Drosophila melanogaster. So, though these gynandromorphs are present occasionally in Drosophila, but it is not a common feature. So this is all about the gynandromorph, a very unique thing. And with this, I have completed this chapter, sex determination mechanism. Thank you.